Welcome to the PhD podcast project from Yale's Graduate School of Arts and Sciences. In each episode, we will interview graduate students in different programs at Yale to learn about the exciting work being done at the frontiers of research. We will dive into the motivations behind their work and how it may impact our lives and those of future generations. I am Alvin, a first year PhD student in CS. I'm excited to talk today with Shoiti,
Hello.
Hello, 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 hello.
a third year PhD student in biomedical engineering.
I am Shoiti Srabunti Halder, and I am a third year PhD student in the Department of Biomedical Engineering.
Awesome, yeah. And what exactly are you researching?
So, my research uses engineered heart tissues to study different cardiac diseases, and my specific project focuses on a certain mutation in a protein called tropomyosin that is mainly prevalent in certain families.
Cool, cool. And so, like, if you had to give an elevator pitch to someone who maybe didn't know much about cardiology or heart biology or anything like that, like, what would you say that you really do in the lab all day?
Yeah, so heart diseases take many lives every year. And a lot of the times, the cause of heart failure is not known. It is not always that the person has a bad diet or doesn't exercise, but sometimes the cause is genetic, as in they have a mutation running in the family that they were born with, and they probably don't even know it until they start showing symptoms. And lots of young people die of familial mutations at a very early age, randomly, even before presenting any symptoms of heart failure. So it is very difficult to identify all these mutations and screen them and understand what this mutation does. In my research, we use engineered heart tissue. Where we make a tiny piece of heart and we can customize it any way we want. So we can insert any mutation we want, we can treat it any way we want with drugs to figure out how the mutation impacts the heart tissue, and also how different drugs can affect or alleviate effects of such a mutation. So it is a great testing bed for different heart related diseases and also different drugs that cure these diseases. And this is better than waiting to test it in animals, which is in vivo. Typically, you have mice studies, but mice are very different from humans.
Yeah.
And they take a long time to grow, not very cost effective, and you are killing a life in doing that. So the psychological aspect of it. So with engineered heart tissues, you just get around all those problems, but still get a lot of good, reliable, physiologically relevant data.
Yeah, I remember when I was doing like undergrad biology experiments and stuff, a lot of the papers that we would review and talked a lot about like experimenting on animals and doing things like that. I remember a few of my friends doing biology when they were undergrads, they actually like got really queasy at those parts. Those were like kind of big turnoffs for some of those people. I think some of them actually like switched majors and stuff because the animal cruelty is always like super surprising.
Yes, absolutely. I could not do it, and I'm so happy that I'm instead in a tissue engineering lab, which also sounds very cool.
Yeah, definitely. I remember when we were going over like the survey responses and everything. That one definitely stuck out to us. I'm a little curious, like where is the lab and like how does it work and all of that.
Yeah, so our lab is on Prospect Street, 55 Prospect, this very newish building, which stands out among Yale's old architecture. It's right across from the cemetery. But yeah, it's a completely a research building, and only biomedical engineering labs are in that building primarily. Yeah, so growing a heart tissue, it also takes some time and preparation, but it's way easier than you would have to, way easier than growing an animal.
Yeah.
So what we do is we actually go to a slaughterhouse and we obtain big hearts from the slaughterhouse. And we bring those hearts back. They sell that to us, I think, for $50 for
$50.
one heart.
I guess
Yeah.
like since people some people eat hearts and stuff, right? So I guess it can't be too crazy expensive.
I guess, but I think they were gonna throw that away. But once they realize there is a demand for that,
Yeah,
they
yeah.
do charge us for that. So yeah, we bring it to our lab and we chop it up. We make nice blocks from that heart. We try to get the left ventricular portion because that's what we are mostly interested in, and then we freeze that, and then it basically is a solid block. We're freezing it at very cold temperature, like -80, and then we slice them, and the slices are very thin, as in 150 microns thin, and we slice them using a cryostat, and then you have this tiny sheets of like heart tissue, big heart tissue, right?
Sorry, just for the listeners out there, what's a cryostat?
A cryostat is a device that where you can mount frozen blocks of anything, and you can take very precise slices
Okay.
from that. Yeah, so then you have this thin films of heart tissue, which then we use a laser cutter to sort of cut out rectangular, two-dimensional pieces from there. Yeah, it's just like, when you would do cutout in kindergarten, kind of like that. But yeah, so once you have those rectangular pieces, we have to decellularize them first. So we want to make sure there is no pig cells in there. So we kill all the cells in that pig scaffold so that all that it's left with is just the structure, but no really living component from the pig anymore. And then we introduce human cells to it.
Okay.
And the human cells inhabit that scaffold, and after you have grown them for a little bit, they start beating. Now these cells can be of various origins, whatever you want, whatever your project needs. But previously we have done cells taken directly from a patient. We actually try to get stem cells from patients. So if there is one single person with a certain mutation, we can just get their stem cells. We can make cardiomyocytes, which means heart cells, from the stem cells. So I know people hear a lot about stem cells. I can explain what that means. Stem cells are cells that have the potential to become any other kind of cells. They're called pluripotent, as in they have the potential to become anything else if they get the right cues. So we give them the right cues so they eventually turn into cardiomyocytes, which are heart cells. And then we put those heart cells into the scaffold, and after growing them following a certain protocol, they will start beating. And then you can essentially characterize what would happen to that person if this drug was given, or if this mutation worsens, or in like 10 years, because in engineered heart tissue time, the timeline is much more accelerated because it's such a tiny strip of tissue. The pipeline is at this point very straightforward to us because we have been working on it for so long. Yeah, so the total culture time from the day you put the cells in to the day that you're ready to test is 2 weeks. So within 2 weeks time frame, the tissues are matured enough and they have been in culture enough for the mutation phenotype to start showing for us to see some changes. Obviously, before this culture period, you are doing multiple things at the same time, like you're making sure you have all the equipments, you go get the heart pieces, you're processing all of that. You're also growing the stem cells. The growth of the stem cells are also something that you have to do on the side. Stem cells take a lot of time to differentiate into those special heart cells.
About how long do you think the stem cell differentiation?
So the stem cell differentiation part is another 3 weeks or so, and then 2 weeks once they're turned into tissues. So about 5 weeks of prep time, but then each round you are doing a big batch, and you could do like 30 tissues each time, or and then also you don't really do one and then wait for 5 weeks and then start the next one. So it's sort of all going parallelly through the pipeline. So we churn out probably more than 100 tissues a month at this point per person.
Wow.
Yeah.
So it's literally like you have just 100 copies of a single person's heart tissue, or like whatever sort of mutation you're looking at, you have a
Yes.
bunch of copies that you can experiment on
Exactly.
with different drugs and things like that.
Yes,
Wow.
exactly.
Okay. That's cool. That's cool. What actually like got you into this whole field as, when you were an undergrad and when you were even younger than that? Like,
Yeah.
how did this all get started?
So I can talk about my high school a little bit. So I went to high school in Bangladesh.
Yeah.
I grew up in Dhaka, and I went to a very good school called Sunnydale. And growing up, I really didn't know what I wanted to do. So until the 10th grade, I didn't even know if I wanted to do science or like business because I liked everything. But it was more like towards the end of that, I was realizing that I really like biology, but then I also really like math. And then the only subject, I did some research, and the only subject that seemed to need both of these two and utilize components from both of these two subjects was biomedical engineering, which is why I sort of strayed away from pure biology or pure math because I wanted to do both. And so I came to the U.S. in 2015, and I was admitted to Columbia University,
Nice.
and I decided to do the BME major. I was very decided that I'm going to do that from day one. And also, funnily, I was checking back at my common application back when I applied,
yeah.
and they asked, what do you want to be in the future? And I did say I want to do a PhD. So I don't remember actively thinking, oh, I'll get a PhD someday in high school, but somehow I wrote that down.
That's amazing. Yeah. Just a straight shot through, like undergrad and everything. You just always wanted to do biomedical engineering?
Yes. So at
Awesome.
Columbia, I joined a lab, which was a lab I really wanted to join even before going there because it just seemed fascinating. So they were also a tissue engineering lab. So like I explained, tissue engineering sort of allows you to test different diseases. It's a testing bed for diseases and drugs and different methods of, understanding the disease progression. And this lab was tissue engineering, but in the field of orthopedics. So they were looking at osteoarthritis. And back then, I know that my grandma was like really in a lot of pain from lifelong osteoarthritis. And I was like, oh, there is no cure for this disease. Maybe I can go learn a thing or two about it. So that was like my motivation for joining that. But then I discovered this whole other field of tissue engineering. And when I was applying to PhD programs, I knew that whatever I wanted to do next, I wasn't married to orthopedics specifically, but I wanted to do something that used tissue engineering to answer these questions because I thought it was so cool.
We're covering that sort of as the main theme for this podcast episode. So if you could talk a little bit more about like your background in Bangladesh and things like that. Moving over here, what that was like. Any experiences that stood out to you while you were getting used to the U.S.?
Yes, of course. So I grew up in Bangladesh. I mentioned in Dhaka, which is the capital city. It's the metropolitan city. So I did grow up in a very protected environment. Dhaka in general is a very unsafe city. It is, it's not safe for children or teenagers even to travel alone from school to home or vice versa. So you always would want to have like someone, a guardian, accompanying you. And both my parents were working. So I come from a very middle class family in Dhaka. It's my mom, my dad, and me and my sister. I have a younger sister. My parents were both lawyers. My mom's a human rights lawyer. My dad's a corporate lawyer. So it was natural, everyone, all my relatives would say, oh, so you're probably going to be a lawyer, right? But I used to feel that black and white is not, I like colors. That was my first aversion towards the field of law. And I was good in science, so I sort of moved away from that.
Black and white. So like, was it always just because your parents would be wearing like really
Yes,
drab
like
suits and stuff
yeah,
like that?
yeah, white shirt and then the black robe, that lawyers typically
Oh,
wear. I know that
yeah.
here I think suits are more common, but in Bangladesh people still wear what we would have worn during the colonial times. So
Okay. I see.
yeah, so when I first came to Colombia, I did not expect to get in because it is very
Yeah, it's Colombia.
yeah, so it was when I did get in, it was nowadays I know that fortunately Bangladesh has more Ivy League alumni.
Right, of course.
But back when I was applying in 2015, I'm sure there were others, but it was so unheard of, or just that in my circle, I just didn't know anyone. So everyone really tried to discourage me. Like, don't apply so high. You're not going to get in anywhere. Just, curb your ambitions a little bit. So I was definitely all of that, but my parents were like, do whatever you want, because they didn't know about this process. They were just experimenting with me, I guess. So when I did get into Colombia, my parents were overwhelmingly happy, and they actually came to help me settle down.
Right, yeah.
And the other thing was, it was the cost was a big factor for me, and it was out of our expectations that Colombia was going to offer a complete full ride, so I could attend, because I know I mentioned that I come from a middle class family, but when you convert that gross income into US dollars with the exchange rate, it is
Boy.
like really even below minimum wage if you're working full time. So so that income allows us to sustain a decent lifestyle back in Bangladesh, but we couldn't pay even a tenth of the educational expenses over here. So I was super lucky that I got that opportunity, and my parents were very happy, so they came to get me settled in. It was hard to say goodbye to them. I remember that when I first came to New York, so this is the other thing. Growing up in Bangladesh, I identify as a religious minority in Bangladesh. So Bangladesh is a country that's majority Muslims.
Yeah.
And my family is Hindu. So so there was some kinds of like discriminations or microaggression incidences that often happened. So my parents were always keen on making me leave the country as soon as I got the opportunity to do that.
Do you have any particular memories of any discrimination, or were you kept mostly away from the worst of it, or stuff like that?
My parents tried their best to just shield me from the worst of it, but I know that my dad was denied the opportunity for a faculty position just based on religion. I know that there were some classmates who would just be rude to me and bully me because I was from a different religion. So all of those things did affect us to some extent. So my parents were like, the situation here isn't getting any better, so maybe if you find opportunities abroad, that will be better for you. But don't get me wrong, I love my country, and I have definitely amazing friends and teachers who really support me, and not everyone is like that, but a lot of people also are. So when I first came to New York, I rode the subway, and there were people of all colors, all fashion senses. There were people with the big afros, people who had green colored hair. Like just the diversity in this one train that I was sitting at, just like looking across the train and just looking at so many different kinds of people from all over the world. And that was the first time I realized that I realized how illuminating and encouraging diversity can be, like the power of diversity, because when I'm one of them, I felt that okay, this means no one's going to, I'm sorry.
No, it's cool. for the listeners at home, Saiti just received a bunch of
I don't know.
phone messages. Very popular.
All right, sorry about that.
Oh,
But
that's
yeah,
cool, that's cool.
as I was saying, so when I was on the train, I just, that was one of a big moment of revelation for me, that wow, this can be, this is what diversity looks like. Because back home, everyone's brown, you don't see a not brown people. And the other terms of diversity is like religion mostly.
Yeah,
So
definitely.
yeah, that's the other criteria. But here, all different races, religions, fashion senses, sexual identification, like all different things. And I was like amazed by that one scene, my first subway ride in New York. And I was like, oh, I don't know, what's coming for me, but at least here I'm not going to be discriminated against. So that was the positive. The negative was the culture shock. Like, I didn't know how to navigate subway. I didn't know how to order a sandwich or a coffee. I would always go up to the barista and freeze because, oh my god, I'm gonna say something wrong and sound stupid. So all of those things were hard in the beginning, but then it all became easier over time. And
definitely.
But in the beginning, I was very, very scared of speaking up in class or in discussion sections, not because I didn't know the material, but just because I was afraid of sounding stupid and I was ashamed of my accent. But Colombia also had a very diverse environment. So there were people, and I had a really blessed to have a wonderful international cohort of students from all over the world. And they all had their respective accents. So over time, as I hung out with them and, as I realized that, okay, this is really, no one else is thinking about this except for me, that was easier to get over.
Just circling back to doing research and just in general, like how do you think all of that sort of filters into engineering heart tissue and doing bioengineering and stuff like that?
Yeah, so definitely both the research I did at Columbia and the research I'm doing now at Yale, both of these are very, I would say, cutting edge. And it's a new technology. It's, trying to address problems that this whole scientific community has been unable to sort of crack into in the previous years. Or maybe, it wasn't something that was of pressing interest. And I know that I was recently looking at this report. So Bangladesh doesn't really have a lot of bench research facilities, or I would say very little, if any, at all. There is clinical research going on, but very few bench research opportunities, even in the best universities. And
What's bench research?
Bench research is when I say bench research, I'm referring to wet lab research. What you do in a lab, essentially a lab where you can do all this cell culture stuff and everything. So Bangladesh doesn't really have that. That academic environment is not there. And that is primarily because a lot of this money that the money behind all this research work comes from the government in the case of the U.S. My work is funded by a NIH grant. NIH is National Institutes of Health. But in Bangladesh, there is not really a central body like that can fund health research. Maybe there are, but even the funding is I'm sure pretty limited. So there is clinical research though, like epidemiology and things like that, but this kind of wet lab research is uncommon. And since coming to the US, I have definitely learned to appreciate the importance of this more. And I was saying, I recently read a report about how many ER cases, emergency room cases in Bangladesh lead to sudden cardiac death. Sudden cardiac death is when a person who never had any heart problems before, no complaints at all, suddenly just passes away because of a heart failure. And this is exactly the kind of thing what the disease I'm working on, which is hypertrophic cardiomyopathy, does. It is the leading cause of death in the young population, and these people just die randomly one day of heart failure, whereas they've never seen anything wrong with their heart in previous check-ins with their physicians. This is because the screening method does not exist. No one thinks, oh, let me go and check if I have hypertrophic cardiomyopathy or anything, right? So this is super important. This patient-specific treatment that scientists are working so hard in the U.S. and also other parts of the world now to promote, because there is no one-size-fits-all kind of treatment for some kinds of diseases. So in Bangladesh, we are far from that. So I was wondering when I was looking at that report that a huge amount of people, they just passed away from sudden cardiac death. And there was no, like, the report mentioned nothing. Like, we investigated further why they just died. Yeah, they were just giving us a number, and I was wondering, I wonder what percent of that has to do with a familial mutation. Like, if they were screened, I'm sure you can do that. And maybe, yes, you may not be able to save that person who has passed away, but you can screen the other family members and prevent the same from happening to them as well. So the importance of personalized medicine is so important in today's world. And we haven't done this yet, but I imagine that in the subsequent years, as this pipeline becomes more and more common, that we can get families or people with this mutation to use our research to consult with their physicians about treatment regimens and plans. So that's the end goal right there, that patient-specific treatment.
Awesome. Well, thanks again for coming. This has been Saiti Halder talking about her work doing biomedical engineering, growing heart tissues. Thanks again.
Thank you so much.
Yeah. See you.
Thank you for listening to the PhD podcast project from Yale's Graduate School of Arts and Sciences. We hope you enjoyed this episode, and don't forget to subscribe and check out other episodes on our website.